A power analysis is a process of finding the correct sample size for your project. If you use too many, then it wastes your time, effort, and money. However, if utilizing too few, then this also wastes your resources and yield invalid results. To cognize power analysis, you would need to understand some parameters. General rule to figure out sample size for any study depends on the effect size, standard deviation or variation in the population, type 1 error, which is also called alpha, or acceptable significance level, commonly set at 5% level, also called 95% confidence. Type 2 error, beta, or power of the study, commonly set either 80% or 90%, with a type 2 error of 10 or 20%, and whether it's one-sided or two-sided statistical test. In statistics, effect size or signal is the difference between the value of the variable in the trial group with that in the control group. For example, if the drop in blood pressure in the control group is 5 mm of mercury and in the trial group it is 10 mm of mercury, then the absolute effect size will be 5 mm of mercury. Effect size is usually of clinical or scientific interest or public health importance. It could be small or large. Variability, expressed as a standard deviation or noise. It is the variation that you capture from previous studies, existing data or literature, and if no study is available, then you could conduct a pilot study. If no good estimate is accessible, then it is still worth utilizing a low and a high estimate Cohen's power analysis to figure out if it makes any difference to the sample size. Cohen's categorized that the value of 0.5, 1, and 1.5 are categorized as a small, medium, or large. It is important to note that variability does not need to be calculated when comparing two proportions. It is enough just to specify the other variables. The significance level and type 1 error. Every researcher is keen to get a statistically significant result from their studies. But if the study is not well conducted and somehow a researcher manages to attain a p-value of less than 0.05, which is basically a false positive result or error, then this is described as type 1 or alpha error. Type 1 error can be controlled by choosing a significance level, which is usually set at 0.05, stating 1 out of 20 could be due to chance, which is quite arbitrary. In certain situations, it is also set at 0.01, shrinking down the possibility of chance, but this increases the requirement of a large sample size. Similarly, if a researcher fails to detect the true difference and gets a non-significant p-value of more than 0.05, then this is a false negative result or error from his study called type 2 or beta error. It is difficult to control type 2 errors. Choosing the study power, that is, the chance of identifying a real effect if it is present, relies on the research question. If the magnitude of failing to detect the effect a type 2 error is serious, such as in life-threatening research, then setting a relatively high power of 90% that requires much bigger groups of subjects would be more appropriate. However, where their type 2 error may not have grave consequences, then a power of 80% may just be enough to hook larger effects, and consequently fewer subjects will be sufficient for the study. The alternative hypothesis may be that the mean could go in any direction with a statistically significant result that is two-sided, or that they differ in a specific one-sided direction, example that the mean of the treatment group is greater than the mean of the controls, which is one-sided. All 5 components involved in a power analysis are interrelated. If any 4 of them are listed, the 5th one can be calculated. For example, if an anesthetist wants to compare the effect on blood pressure of 2 drugs for patients undergoing gastrointestinal surgery. The age of patients was between 40 to 60 years. Mean systolic blood pressure was 141 mm of mercury with a standard deviation of 36 mm, which is a noise. The essential parameters required for the calculation of sample size are a difference in blood pressure of 20 mm of mercury, which is effect size, or more would be of scientific clinical significance, and it's not a statistician choice. A significance level of 0.05, a standard deviation or variability of 36, using a power of 90%, and a two-sided t-test. Then the signal-to-noise ratio would be 20 divided by 36 equals to 0.56. Using statistical software packages, the output estimated the sample size of 68 per group, giving a total of 136, which sounds like a lot. The same anesthetist now decides to do the same study but restricting the age band to 40 to 45 years. These age groups had a mean blood pressure of 108 millimeters of mercury with a standard deviation of 9. Assuming a 20 millimeters of mercury difference between groups would be of clinical importance as before. With the same assumptions, the signal-to-noise ratio is this time 20 divided by 9, which is 2.22. This is only 6 per group with a 90% power. So, by using uniform age bands, the number required is reduced from 136 to just 12. In real life, however, restricting or shrinking down to a uniform group can compromise the generalizability of the study findings. A researcher can then, during the design of a study or planning stage, make pragmatic choices like switching from parallel randomized controlled trial design to randomized block design or factorial design that would answer the research question without compromising the validity of the study. Some more factors that can be considered while calculating the final sample size include the expected dropout rate, an unequal allocation ratio, and the objective and design of the study. Different types of formulae could be for one sample mean, proportion, or percentages. Formulae for two samples, like differences between two means, two proportions, or two percentages. It also depends upon the study types and designs. For example, calculating the sample size for multiple regression analysis, the more predictors, the more sample sizes needed.